Thank you so much to the students and the organizing committee for having me. I always look forward to this. I've actually missed it the last 4 or 5 years because it's been scheduled the same time as the North American Plant Phenotyping Network, which if you haven't been to, is probably the most exciting group in science today. I'm about the oldest at 45 when I go. It's all graduate students, postdocs, assistant professors, and we've gone from, here's the way I'm collecting this data to just groundbreaking discoveries every presentation now. So I'd encourage you to go to that meeting, but it's online this year. It'll go back to person next year. The et al. Here is doing a lot of work. I'm going to highlight some of my contributors. I was hoping to update the acknowledgement slide, but there's just way too many people. So I'm sure I missed some people. My apologies, but hopefully I highlighted a few key people in here. I'm going to do something different. I've been given the same presentation for a couple of years, and I really wanted to challenge myself to try and present this in the way that I've been talking to. For instance, I was just at the American Association for the Advancement of Science meeting, and most of them don't care about plant breeding, right? How do you convince them? How do you convince the physics community that looks down upon agriculture that what we're doing is impactful and important, right? So this is generally the way I've started introducing stuff. So for all of you, just keep in mind, if you have a sibling, or an identical twin, right? You look similar, you share similar traits, your intelligence, athletic ability in general, it's not perfect, your body shape. But how would you describe that difference, or how you would describe that appearance to a stranger who's never seen you or never seen your photo, right? We tend to lack the language to do that. We'd say something like hair color, but that doesn't really describe anybody in particular, or height or skin color. Maybe there's a distinguishing feature like a neck tattoo or something, maybe that would help. But in general, we don't really have that concept of that language and ability to do that. However, we have evolved this ability to recognize these relationships, right? We can recognize relatives. Subtle similarities. So think about somebody with Down syndrome, for instance. How would you describe what somebody with Down syndrome looks like, right? It'd be a little bit challenging, but we can recognize that, right? My father-in-law, when he first got Alzheimer's, we could tell years before his diagnosis, both in terms of his speech ability, in terms of his facial features, in terms of his mannerisms. There are lots of things that we don't have language to describe, but that we can perceive. Now, when we think about humans, there's a lot of focus on finding genetic determinants of all these things. But humans have a limitation that plants don't. So let's think about your sibling or twin again. You grow up in the same house, you're going to look similar, you're going to have those similar features. But then imagine you and your twin are separated at birth, and your twin is raised in a war zone. All those relationships kind of disappear, right? The stress and the food insecurity temporally throughout that time that twin is raised in that war zone, that different environment, that different G by E, they'll be shorter, they'll be lower IQ, and they definitely will look different from all that embodied stress and lack of nutrition. So this is my family here, right, my sister and I, and you can see we look similar. We look similar as kids and we look similar as adults, but we don't look the same as kids as adults, right? And there's various measurable phenotypic features, both as children and as adults, that demonstrate not only our genetics, but our interaction with the environment as we're growing up, right? And this is the thing that's really difficult for us to think about and especially to measure. Now, facial features are known to be heritable. You can actually measure those, but so far nobody's really taken an interest in it. It's very difficult to do. Goats also have face recognition. They have resemblance of relatives just in the face. So the first time I got interested in this concept was actually with Natalia's PhD. We were looking at some NGRDI data, which is a vegetation indice. And days after planting is on almost every x-axis of the slide, so we're trying to measure things throughout growth. And NGRDI is just a ratio of 2 visible bands of light. So if we look at the various hybrids up there, those 7 hybrids, 3 of those are commercial checks from different companies, so presumably unrelated. The other 4 hybrids all have a common tester, TX714. And if you look at the pattern of NGRDI throughout the growing season, you see that those individuals that are more closely related have a more similar pattern, right? Very similar to what I was just showing you with humans. And that pattern is not the same at 20 days after sowing or 120 days after sowing, right? There's different phenotypic information in there, but they still maintain the same resemblance at any time point through there. Now I want to talk just a minute about genomic methodologies. So when I started in graduate school, GWAS was becoming really big. So did one of the first studies on GWAS and sorghum. Before that, in 1986, we had QTL linkage mapping. Patterson and Buckler and Bernardo and Yu each made huge contributions to these methodology. But think about the mentality shift it takes from going from this idea of identifying genes and that we'll scientifically use those genes we identified to improve our crops, which is the QTL mapping or association mapping methodology, to this methodology that everybody here seems to have shared today of prediction. What's beyond that, right? Where are we going beyond that? I think where we're going beyond that is not just focusing on genotype, but focusing on phenotype, which I think you've already had a lot of great introduction to. Now, I personally grew pretty frustrated with QTL mapping and association mapping. We'd find loci, and then they wouldn't be replicated across environments, as we heard. There's this problem that the human geneticists have been talking about for over a decade now called the missing heritability problem. And it's quite simply that if you take all the loci from all the genetic mapping that you do in a population, it's always less and often substantially less than heritability as you measure it in a pedigree or family type relationship. And the question was always why. And the human folks really focus on these top ones. But I think the important part here and why my search for a different way to approach plant breeding and plant science is because I just keep on hearing people say, well, if we could just sequence more individuals, if we could just sequence more markers, right, we'll have a better result. And still the missing heritability problem remains. So I'm going to read through this list. I think it's one of my only all-text slides, but small effect loci, big problem. I'll show you that in a minute. Low frequency alleles, especially that are stratified across populations. Population structure soaks up a lot of variance if you make GWAS populations with a bunch of diverse material. Structural variance, there's a student here at Trevor at A&M looking at this in Drosophila. Looks like it might be sort of important. Context dependency, which includes some of the below. The environment in G by E, we can measure that really well in plants. Humans, not so much. The biggest driver for me, though, and the biggest source of frustration is epistasis. We just ignore it. And Trudy McKay has a fantastic article that convinced me that because of epistasis, most genetic effects will never be knowable. We can estimate them in a certain genetic background, but you put them in a different genetic background, I've seen the effect increase, decrease, be neutral, or even have the opposite effect. And it made me sort of give up on this idea of identifying genes and using them in my breeding program. Epigenetics seems to be an important one, and of course just emergent properties, which I'll sort of try and tie to in the end. A great example of this that we had in plants recently, this was part of Alperedak's PhD project, was looking at a disease lesion mimic. So these are little spots that get in the field that are hypersensitive response. There doesn't need to be a disease present. So he did both genomic selection and GWAS. And then what we tried that was a little bit unique was this was a quantitative lesion mimic. There was only one candidate in GWAS. There weren't any even really close peaks. But I said, well, let's put the rest of those markers into a model for genomic prediction. And so if you look at A and B, if we include the whole set of genomic markers, 39,000 SNPs, we soak up most of that error variance. It can explain most of the variation going on there. If we use the subset of genomic markers that were the top, I think, 50 hits in GWAS, they weren't actually significant, so nobody would call them. We get more error, but we still explain a lot of the genotypic effect. But then as you go over to prediction ability, using that subset of markers, those 50 additional loci we never would have called in GWAS seem to be really important in prediction. That to me told me that there was a lot of epistasis going on. Probably also because we had this stratified across populations from genetic background effects. So even though this is something that people consider a single gene mutant, we find that the epistasis is pretty damning for actually trying to use this in a predictable way. Okay, so why am I telling you all this and starting with humans? I tried to introduce this with the idea that I think plant science, 1% of people are involved in agriculture now. We aren't seeing huge growths in the number of plant breeding programs across the world. Plant breeding has to make a case that we can lead in some areas across science. So I often will explain to other scientists, Gregor Mendel, working with plants, basically founded genetics. Ronald Fisher, working with crops, basically founded statistics. And then I have this example down here too. Hopefully everybody knows Lysenko, who put Vavilov in a pretty bad situation. Vavilov, one of the greatest plant scientists alive, plant explorers alive. So Lysenko and then Charles Davenport was implicated in a lot of scientific racism. So as I talk about human issues, I think we really need to make sure that we're not creating those mistakes of the past. People will take some of this information, as we've already seen happen, and use it in terms of scientific racism or ways of excluding other people. We need to just be cognizant that that can happen and make sure if we lead it, we can grow huge populations, we can easily measure them temporally over life cycles, and we don't have ethical concerns. But as we extend these methods to humans or even animals, we seem to have a little bit more chance of our methods being perverted. Okay, as we think about phenotyping and phenomics, I think it really fits well into the continuum of advancements we've seen in plant breeding. So in the background there is the US corn yield, and everybody's probably seen this, open pollinated varieties, hybrid vigor by Shell and East, Henry Wallace starting that first seed sales, going to double cross hybrids, single cross hybrids, and modern hybrids. And what we've really seen is a transition from the breeder's eye, right? And what I showed you with people hopefully connects with those of you who don't do plant breeding to think about the breeder's eye. I know Bill Rooney can go out in the field and look at a plant and know that's pretty good or that's not going to work, right? But there may not be a measure that he has. He just sort of knows from the accumulation of features. So it's been called a prophetic gift or a trained eye. We've been moving towards objectivity. I don't think we can ever get to full objectivity because we don't know what next year's weather is going to be like. We don't know what next year's economy is going to be like. But still, for the parts we can control, we're trying to make them more scientific and more objective. And as we think about this in the context of phenomics, so genomics took 30 years to mature. I think we've really only been working on phenomic methods for the last 5 years. I mean, we started the UAS program here at Texas A&M 10 years ago, but it really took a while to get into the phenomic side of things. So I think the biggest discoveries, for those of you who are graduate students, are about to occur. We think about the creation of a genetic map, right, in genomics. That was something that the software, whoever first designed some of those first software packages, were extremely highly cited and influential in their activities. I think there's going to be quite a bit of opportunity in the phenomic space for those same types of things. So moving into high throughput field phenotyping, there's 6 main ways I think about this. The first one, which is what everybody starts with, is automating routine measurements. So I'm going to talk about plant height. I'm not going to talk about estimating grain yield directly or estimating disease directly, but there's lots of people working on this. Measurements are really laborious to take in the field. If we can automate them, it's going to save us resources. We're going to be able to scale our programs. That's fantastic. I'm more excited about finding new signatures of lateness. So I'm going to talk a lot about temporal growth patterns. I'm going to talk a little bit about senescence near the end. And then we move into phenomic prediction. And I'm going to talk a little bit about some examples of phenomic prediction. And then follow, sort of at the end, talk a little bit about AI on images. I think all these things that we discover are going to be new measurements, probably that weren't known before. So these things can also be used for helping farmers manage stress. I think they're also going to really open up opportunities in finding mechanisms of biological importance that nobody's really measured before. And how is that? Let me give you an example. So repeatability or heritability, I think, is the key measure for any type of new phenomic or high throughput phenotyping thing you could measure, right? But the way that most people have approached this, at least our group certainly did when we started, is we say that there's some truth. And let's be clear here, there is no such thing as truth when you're making measurements in the field, okay? There is no ground truth. You're making a ground measurement, but that's not the true value. It's another estimate. It might be a good estimate, it might be a bad estimate, but we make some ground truth, ground measurement, and then we had the aerial vehicle, the UAS measurement on the y-axis. And so you can see that they're pretty well correlated, but the ag engineers said, well, see, this isn't as good as the ground truth measurement, right? But how do you know that? There's no possible measure there that could tell you which one of those is better. It's a simple correlation between two probably poor measures. It doesn't tell you which one's better. But when we go over to measuring heritability, what we saw was that our UAS measurements, or our drone measurements, sorry, I'll use those words interchangeably, oftentimes were as good or higher in terms of repeatability than our traditional ground measurement. So to me, that says the UAS measurement is a better measure. But I think overall, any type of new tool should be used to not just increase the throughput of what we've done in the past, but actually question it and say, can we measure something better? Can we measure new things? So I'd leave you with that last part there. How accurate is our manual phenotype training data? And I'll show you an example at the end where I think this drives a lot of it. We trust the combine, right? But the combine isn't always right. Combine has errors. People out with a measuring stick have errors. So we can extend this into near-infrared spectroscopy. So near-infrared spectroscopy, I don't know if it's really been introduced today, but we use this Thermo Fisher Antares. We scan whole kernel corn or whatever commodity you're interested in. We grind it, we scan it again, we send it for chemical analysis. And what you're seeing on the right is an absorbance spectra. And various chemical bonds absorb different wavelengths of light, and we can use that to say how much protein there is or how much starch, right? We develop a predictive equation, and then we can use that to scale our measurement. So we usually only measure 10% or less with wet chemistry, and then we can basically, for free, some undergraduate time scanning these things, extend that to an entire breeding program. So I'd like to show you the example at the bottom. On the x-axis is the actual value measured by wet chemistry for crude protein and phosphorus. On the y-axis is the predicted value from that IR. And so anybody would look at that and say, oh, crude protein is a great measure. Phosphorus, not a very good measure, right? The interesting thing is once we extend that to our entire breeding program with multiple locations, multiple replicates, right, much larger population sizes, we see that the genetic variance for phosphorus is almost as high as the genetic variance for crude protein. But surprisingly, the error, the unexplained error in the population is lower, right? So this led us to do some simulations. Holly Lane took some code that we do in my graduate class and really extended it, did a nice job of showing and coming up with the conclusion that high throughput can produce better decisions than high accuracy. So we simulated additional error with the high throughput phenotyping methodology, including up to 10-fold more error. But if you can screen double the number of plots or 10 times as many plots, you substantially increase your ability to make decisions when you do know the real truth. In this case, the truth was simulated, so we actually do know the truth. So that sort of explains how some of these predictions, some of these shortcuts to measure something like protein through NIR can actually be more predictive just because we can scale that measurement to a much greater level of population. I'm going to show a little bit more on height first before I switch to some of the other measures. This is a little bit of an older study from Stephen Anderson showing two different drone platforms, a Tough Wing and a DJI Phantom 3. On the x-axis is days after planting, and so you can see those growth curves. So in maize in Texas, this doesn't apply to the Midwest, but in Texas, plant height is highly correlated with yield, and I think it's just a measure of how resilient it is to our stresses here. And what we found was the manual plant height measure was 0.23 to 0.28 correlated with yield, whereas the UAS measurements or drone measurements were much higher. More importantly, we're taking measurements throughout the growing season, which we never could do with rulers before. And thankfully, we had this information when Alper looked at these isogenic lines we created. So we created some isogenic lines with a locus from a GWAS study that was discovered in hybrids in our program. And we created those isolines in inbreds, and he wanted to see if we could validate those two loci for plant height, which also worked for yield in this case. And what he found was really surprising, but a happy accident, that using manual measures and measuring at the end of the growing season, which is what we did in the hybrids, the differences were non-significant. But because we had all those early season measurements, we learned two things. First of all, yes, it was significant, and second of all, it's actually occurring far earlier in growth, whatever these two genes are, than we expected. But the problem comes, how do you make this, these UAS measurements, especially when there's so much data, into something meaningful? So to date, I think Jacob Washburn is the leader in a publication, not necessarily the leader in collection, but the publication so far in the number of flights. He flew the Genomes to Fields trial 40 times, over 1,000 hybrids. And the reason I wanted to show you this slide was because of the Weibull fit. So all of these measurements independently are really difficult to make sense of. But you can fit a line through these and actually develop a 3-parameter predictive equation that can then model this. The nice part about that is it takes a lot of the noise out. Each UAV flight is a little bit noisy. But by having so many UAV flights, we can fit a best fit model and then use the parameters of that model in GWAS, in genomic prediction, whatever you want. Okay, so I talked about plant height because it's sort of something everybody can connect with. It's pretty easy to think about and measure. All the rest of the traits are much more complicated. So I showed you people and talked about how we recognize resemblances and how we might measure them. Well, we've done a really good job of saturating the phenome. Over my 18 years here, I just look at 2013, 680,000 markers. By 2022, 1,500 inbred lines with 12.2 million markers. And in 2023, 46 million SNPs on 1,200 samples. Right? So we've saturated the genome in maize. And what does that mean? Saturated genome means if you know if you collect a new marker, there's no new information compared to the other markers it's linked to. So we really can't discover many more SNPs that add information in this case. Structural variants, I mean, there's some limitations. But so far, we haven't talked about saturating the phenome. How many measurements do we need to make before each additional measurement we make can be described by other measurements? So once we saturate the phenome, I'm pretty certain we'll be able to predict a lot of different things we've never been able to predict before. And what will that take? What it'll take trying to figure out how we're going to make the maximum number of measurements. So language sometimes gets in the way of this, as I started showing you with phenotype. So a phenotype is plant height. A phenotype is a specific vegetation indice. Phenotype is a totality of plants' observable characteristics. And we usually believe it to be of interest, which is why we measure it. But just because we measure it, just because people have measured it for the last 50 years, does not make it a good phenotype or a useful phenotype for a farmer, for a breeder, for a consumer. A phen is a more elemental component of a phenotype, but the phenome would be the totality of a plant's measurable characteristics and estimable physical characteristics. And that would mean across environments. So if we measure something in Texas, can we predict what its phenotype would be in Missouri? What will it take for us to be able to measure the phenome or to saturate a phenome? I also need to point out that phenomic selection is not the same as phenotypic selection. I've had a lot of people really confused that what we're doing is making selections in the field by looking at the plants. The goal in phenomic selection is objective measurements in enough dimensions that capture the actual phenome. Also, importance of language, these get used and interchanged a lot. I've actually had an example where there was an article published where somebody said they were the first to do something. I said, this was done over 5 years ago and multiple other people have done it. And it turns out that they searched one of those terms and not all of those terms. So different groups are using different language to describe this, and that's nothing new. Multidimensional scaling and principal component analysis are nearly identical. They just developed in different disciplines. But just be aware that these words are pretty much interchangeable. So as we think about saturating the phenome, how are we going to capture that? It's all about dimensionality. So the benchtop nearest, and I'll talk about phenomic prediction a little bit later, is a spectral way, but you're only measuring it at one time point. Temporal throughout growth is another way. We do that with drones. There's other ways to do that. And then spatially, I think you heard Nick's talk where the spatial resolution of those images is important. Or if you think about phenotyping at an individual plant level versus plot level. The other thing I need to make sure I say is all of this is inexpensive, relatively, and readily available compared to sequencing. So when I think about students from the developing world who are looking to apply tools in their program, if they learn the latest and greatest genomic selection methodology, but they're going back home to their country without those tools working on a minor crop, they're not going to have those abilities. With drones, you basically can create these things within your own program. And I think that's a huge advantage. With a $3,000 drone and a $6,000 computer, you can probably make some real breeding insights from it. So I'm going to talk briefly about some of the evolution of our UAS data phenotyping pipeline, or at least the current pipeline, and this was given to me by Mustafa, who is the world's expert on processing this data. Basically, Aaron, who is now the expert in our program of flying drones, I think he flew over 100 flights in cotton and, 70 or some flights in maize last year. You place ground control points as long as you don't have RTK on the drone. You fly the UAS to collect the pictures, download them, orthomosaic them. These guys are using Metashape. Pix4D is also an alternative. Generate shape files to outline each of those plots. mask the soil and do some other vegetation indice extraction, which is being done now with Python packages. We were using field image R. And then from those vegetation indices, then we can analyze the data as a typical breeder would, okay? And I use JUMP or use R or whatever you need to use. I like to show this because this is one of the biggest barriers that most people have when they first start using drones. When we started our UAS project here, we were told, well, you just draw boxes around each of your plots in an orthomosaic. And I said, that's great, but we got 10,000 plots. I don't really want to draw 10,000 boxes. So this RUAS tool software that Stephen Anderson led, you can put your breeder field book in there, and as long as you have an AB line on the side of the field, it will actually calculate the shape files that you can then extract through GIS very quickly and easily. And it keeps all your connected information, such as barcodes or pedigree. So that's a helpful tool. After we do that, what do we get? And this is from Field Image R, but we get all these vegetation indices. So in RGB images, somewhere in the neighborhood of 30 to 40 vegetation indices. In multispectral, 70 to 90 vegetation indices. And each of these vegetation indices has some unique and useful information for the most part. Some of them tend to be correlated, but we still find unique value in each of these. It's a really short, quick and dirty way to make a lot of phenomic features and increase that dimensionality. What do these look like over time? So again, x-axis is days after planting. These are all just various vegetation indices. And what I'm showing you here is 2 years, 2020 and 2021. You can see that those vegetation indices do not align. And this has been a huge problem. How do you tackle making those predictions when your year-to-year measurements are not even on the same scale, maybe not collected on the same day? And that's one of the big challenges of phenomics. I think Aaron and others are really helping to address and answer that. But it's not like plant height, which you can model in the same way in every environment where there's a simple, nice growth curve up. Vegetation indices fluctuate around a lot. So you sort of need some ways to dissect those. So the first time I got really excited about vegetation indices was with Alper's study here. So what this briefly showed, again, x-axis is days after planting, y-axis is all the different vegetation indices. The highest predictor of yield actually occurred between days 24 and 63. That's before flowering. That's completely the opposite of what any physiologist I've ever talked to will tell you. My hypothesis on why this is that breeders have gone out to the field in maize looking at plants after flowering. And we've selected most of the useful variation there. So what this is really showing is where there's variation that's useful to select, right? This is where there's still variability. There may not be very much variability after flowering in these lines, but there's a lot of variability when those plants all look identical to us, but they don't look identical to some of the phenomic measures that we're making. So that was pretty exciting. Similarly, with Aaron's paper that he worked on with Alper, RCC, a vegetation indice, the most predictive of senescence 17 days after planting. Right? Predicting the senescence of a plot 17 days after planting. You know, if you accumulate all of those vegetation indices, you'd get a much better picture. But I think one of the real advantages of some of these phenomic measures are you don't have to wait till the end of the season. A lot of them could be done in real time as the plants are growing. And especially looking at early things early on in the season that predict end of season traits. Now I wanted to highlight some of Mustafa's exciting work here. All the vegetation indices that we extract, we exclude the soil. Why? Because soil would dilute the vegetation indices. We're actually interested in looking at head-to-head comparisons of genotypes within the field. Vegetation indices with soil also confound canopy cover with plant health. So by excluding the soil, we hopefully will get more pure phenomic measures of the plant, and we can add canopy cover as a separate factor in that. So the default in Field Image R is this excess green, and you can see it does a pretty poor job of actually masking the soil. So Mustafa identified, and with Aaron I think, identified that hue was a much better VI, and so now those images can be a lot more clean for extracting VIs. The other really exciting advantage that was recently made, and I still don't know if I believe this number, but it's what I've been given, was this new Python pipeline that Mustafa and Aaron and I think Alper and some others developed. 18,000% faster than what we were previously doing. right? Because the processing of these orthomosaics and then extracting the VIs is a real bottleneck. And I'll show you some numbers here in a minute what that means. But by switching software and by switching some processes and by having somebody who's really talented in this, an 18,000 faster method, I think it went from a couple days to a couple minutes. Now, why does that matter? And I'm just showing this slide so I can point out and give shout out to Caleb Buenaventura, who just started with us as our new field tech, and also to talk about scale. This is back 9 years ago from our old UAV project, and I just briefly asked Rooney and Dr. Ibrahim, how many plots they grow. We're talking about 10,000 plots in my nursery, 17,000 plots in yield trials. That fluctuates a lot from year to year. But that's the level that we're able to make decisions on by using drones, right? But we need to scale our measurements to be able to do that. And so a faster Python pipeline can really help with that. Rooney's program, Wheat's program, that'd be a lot of computational time and resources if we were processing all those plots temporally throughout a growing season. So I'm going to switch gears a little now and talk about the Maize Genomes to Fields project. I know, I think it was Diego who mentioned it. This is probably one of the largest public sector collaborations in the field of plant science. We're in our 10th year, 180,000 field plots, 2,500 plus corn varieties, 162 unique environments. All these public sector maize people all collaborate, primarily collecting basic agronomic traits, weather data, but a few have flown drones. And I'll just point out, we just had our second Genomes to Field Genotype by Environment Prediction competition. The winner from last year came in second place this year. But all that data is free, publicly available, and used for a competition. I hope some of our students will be able to enter that in the future. So hopefully you all stopped by Fatma's poster. So she's been using some of this Genomes to Fields data. I point out 2020 and 2021 because we've processed this data for UAV for those locations who had it. But in Fatma's case, she's just using simple genomic prediction, as I'll show you in the next slide. But you can see the range of yield data across these environments from Ohio on the left with a grain yield of 14 to our late planted trials in Texas where we try and get the maximum amount of stress with a grain yield of 4 tons per hectare. So quite a bit of variability, and repeatability was quite high on yield in this data set. So one approach to improve prediction is to include environmental cofactors. And so one of the things Fatma did was found a photothermal time as a predictor, found a range of that photothermal time that really worked to explain each environment, calculated some reaction norms, so how do those different lines perform in those different environments, and then used that in genomic predictions. I think that's going to really improve genomic predictions quite a bit over the long term. The question is what the phenomic data will look like compared to that. So here's an example of the phenomic data that we have for 2020 to 2021 G2F. And you don't need to look at anything in specific here. Just note that there's a lot of different locations, this environment. Variable numbers of flights from Missouri with 87 down to Nebraska with 8. Some collected RGB, some collected multispectral, some did not have plant height that could be estimated. But then go over here and look at these file sizes, right? We're looking at terabytes. And especially when you start thinking about doing multi-locational models, terabytes and terabytes of data. And now Mustafa is part of the CIRCA program, and we're processing CIRCA data, and the drones have improved. The questions have improved. So now we're talking about more flights and even more data that needs to be processed. So this is part of the reason it's really important to have faster and faster pipelines, just as genomics did. I remember when we first were doing SNP calling in the lab I was a graduate student on, and the ABI software would sort of give you an indication, but you'd have to look at each SNP and be like, is that an A or a T, and squint your eyes. We need to get past that in the phenomics side of things. Okay, so this 2020 and 2021 Genomes to Fields data, there were 220 inbreds, and they were crossed by 3 different testers. And those were sort of segmented throughout the US. One of the things you can see here pretty quickly, these are the locations that flew drones, is that these drone flight dates don't match up, right? So that's a really difficult problem compared to the genomic problem of we have the same SNP at every location. So I showed you, this is for temporal plant height, and I showed you one solution as you model it. Those parameters can then be used across environments to be compared and be put in genomic prediction models or GWAS models, what have you. But you can have some really fascinating insights from this. So this is the first time I think anybody's been able to show this from that 2020 and 2021 G2F data, the rate of growth in the different environments. And our late planted Texas environment, the rate of growth was just phenomenal. Those plants just explode out of the ground. It's so hot at that time of year compared to all these other locations. And one of the things Alper's found with this data is the rate of growth across these environments is directly correlated to yield. If those plants grow really fast, they're not going to yield as well. All right, I'm going to switch gears a little bit and talk about phenomic selection. So phenomic selection was first described by Rinson et al. They used near-infrared spectroscopy, which is really nice because you don't have that problem of misaligned data. With near-infrared spectroscopy, every sample has exactly the same wave numbers that have been collected. So they used this in both wheat and poplar, and they found it was as accurate as genomic selection, but cheaper. We happen to have a study at the, from 2020 and 2021 on a hybrid panel GWAS that had been scanned into NIR. Another great use of phenomic data. You can go back and look at it and use it for new purposes that you never intended. And Holly Lane, working with Jose Crosa and some others, confirmed that indeed we had pretty high prediction ability from this NIR phenomic data. But it left me with a big question, because we could also predict the yield just from the NIR predictions of starch, oil, and protein, about 20% less than this. But are these predictions based on some type of composition? Or are they based on some type of relatedness, like genomic markers? And to date, I still don't think I have a great answer on this. I think it's picking up some biological features, and I think it's picking up some relatedness features. This was one of the most exciting studies that I think I've ever gotten to be a part of. I challenged Alpa with this, and he had all this temporal phenomic prediction data. Now this is just from Texas, 1,200 plots, 280 hybrids in 2 of our environments. There were only 158 hybrids with genomic data, used a 70-30 training testing split, 153,000 genomic prediction markers, 1,068 multispectral phenomic features. So you take those vegetation indices by time points, and 525 phenomic features by time points. What you can see is genomic prediction, multispectral phenomic prediction, and RGB phenomic prediction perform similarly. Depends on which CV you're looking at, but they perform comparatively. I think one of my big takeaways, though, was that the RGB data was less predictive than the multispectral. So it could be that the multispectral is picking up more of the biological importance, but I think it really has to do with this. How many phenomic features are available? Look at how many genetic markers we have. Compare that to 1,000 phenomic features. We need to be in the order of 100,000 phenomic features, in my opinion, and saturating the phenome to actually make this a phenomic prediction a clear winner. So hopefully you've visited Aaron's poster. So Aaron's extending this now to that entire Genomes to Fields 2020 and 2021 data set. Because of some of the balance issues, he's doing leave one environment out analysis. He's using 33 RGB vegetation indices. So he has 399 features, but he reduces them to 4 functional principal components. So he and Alper have really been leaders in using these functional principal components to be able to line up data across environments when the drones aren't flown at the same time or have the same development time point. He's run all these different models with NOAA's convincing. There's been some weather data and some environomic models in there. But so far, these are preliminary results, but the results to date is that the genomic and phenomic was substantially better than just genomic or just phenomic. But I think this is really exciting to be able to now scale these predictions to multiple environments as genomics has been able to do for 10 years or so. When we looked at the environments and which environments were predicted poorly, I mentioned TXH3. This is a really high stress environment, very poor prediction ability compared to these other environments. And so I asked Aaron, what's the heritability in each of these environments versus its predictive ability? And it looks like the heritability of the yield trait is really correlated to its ability to be predicted in leave one out analysis. And so that really suggests to me, again, that maybe grain yield, as measured by a combine, is not going to be our best measure in the end for which plant is going to grow best for farmers in future environments. Okay, let me try and synthesize a little bit what I just told you into where this might be applicable. So, if you're working in a purely additive world with only genetics, reality equals theory, I think biotech and gene editing are going to work fantastic. Most of the traits that I've been interested in tend to be really complicated. It may be that there's these additive and epistasis or genetic background effects. It may be that there's a lot of G by E. As we move up this, I think the phenomic prediction and selection is going to really work well where reality is messy, where things are really complicated. And I'm going to show you why I think that is. So we have this central dogma. Most of us were trained under the central dogma. We now know that central dogma has, some challenges. Post-transcriptional gene regulation networks, histone modification, there's signaling protein networks. All these things are impacting the DNA to phenotype type transition. So what's the model of why the phenomics approach might work? Well, I think most biology is emergent. We have all these various nodes, right? And each of these things, if you were doing metabolomics, they'd be almost impossible to measure. But they're all a function of the DNA of that individual, the environment, the G by E, the management, microbes, stochasticity, all types of complexity. But all that complexity impacts the entire network of that organism over its life. So instead of choosing something really hard to measure, the easiest thing to do, I think, is to measure all the easy things that are to measure. So in our case, that's vegetation indices, right? But you can imagine in the future, there'll be other easy things to measure. Right now, RNA-seq is really expensive. Metabolomics are really expensive. Flying drones out of the field is really, really cheap. So we can get all these phenes, and then we can put them into machine learning models and use that to predict the difficult phenotype we care about, right? And that is, if you think about phenotype, it's the great integrator of all biology. The phenotype is the ultimate integrator of all biology. And we don't have to worry about individual markers because we're looking at the entire gene networks that are affecting that organism over time and over environment. Now, where is this going to be important? We keep talking about breeding for yield, breeding for yield, breeding for yield. Corn prices are in the tank. I don't think there's any less starving people, so I'm not sure if breeding for yield is my current goal. But maybe reducing input use, right? Human health and nutrition, farmer profitability, ecosystem service provision, climate change resilience, and flavor. All these things are going to be increasingly important as we think about a society where 1% of people farm, possibly less. And so one of the things we're doing to address this is in the CIRCA project. This is more just a way to explain that CIRCA is a really large project focused on changing nitrogen. Cody Kettler and Mustafa are both funded under that, Dr. Okamoto and Alexa, and then Ed Buckler is the PI of the project. This project's really interested in how do we reduce nitrogen outputs off farm, nitrogen losses, reduce nitrogen inputs. Nitrogen is really a big driver of a lot of the both cost and environmental issues that we see in growing corn today. So one of the areas that we're trying to attack this from is in senescence. So senescence is measured by a breeder. You go out in the field, you score the plant for senescence, right? That alone is really laborious. And then you think you got to go out there to actually look at the senescence over time. One of the things that Alper's figured out here is that he can actually model senescence with this equation. So you fly the drone, the data looks noisy, you can fit a model that fits pretty well, and then based on that, we can actually compare all genotypes at the exact same percent of senescence. We can say at 85% senescence, right, what is, in Dr. Okamoto's case, what is the RNA profile of this root? One of the important thing here, though, is that senescence can be really driven by the days to silking or days to flowering. And so subtracting out the days to silking or days to flowering allows you to adjust for maturity and worry about the grain filling period. And what we find is that's pretty highly correlated to grain yield, right? How long that corn has to fill that ear is going to be directly related to grain yield. Okay, my last few slides are just going to be about AI. I'm just going to shout out a couple projects of using some specific AI. So Cody was leading this, deep learning for senescence. So 22,000 plots. You think about all those G2F locations. We want to score senescence on all of them. That's really laborious, as Nick said earlier, for somebody to sit there and score them. And as Chloe knows, if she's still here scoring plots right now, it takes a lot of time. So Aaron and Cody and Luke, worked together to develop a pretty nice senescent scoring prediction using deep learning, a nice use of deep learning. Nick, you heard his talk, looking at using deep learning to score, oh, I didn't change senescence. You're working on senescence now, congratulations. Scoring tasseling. And then Chloe has been working with Fatma to use YOLO and some other things to calculate leaf area index for the Circa project based on some images that aren't optimal, right? If you have images, can you estimate something like the leaf area index for physiological models from those images? It'd be really laborious to do this any other way. So hopefully you got the chance to see Chloe's poster. So last slide here, why not general AI? So we're taking all these images of fields, and why can't we just throw all that into a model and just predict yield from that, right? I mean, maybe we can. But I think there's some things about general AI that we may be overly hoping for. One is that it's unconstrained, right? So with unconstrained models, you're going to need more parameters to sort of develop some guardrails and some constraints. So you're going to need a lot more data. Most of what I've been talking about, I think there's an opportunity for putting some biological understanding in there or gaining some biological understanding. Which VIs at which time point are driving most of these predictions, right? And then we can go in and investigate them in the greenhouse or do something a little more specific. With general AI, when you're just training something, it's specific to that data set. We don't know what's happening, and because it's unconstrained, you can get some pretty wild guesses. So just as an example, there's this online software, What Breed Is My Dog? And I threw in 2 pictures of one of my dogs, same dog, 2 different pictures, and I'm sorry they got cropped. And you can see what these predictions are. They're all pure lines. He's a mutt. Rhodesian Ridgeback, American Staffordshire Terrier, Great Dane. Different picture of him, Rhodesian Ridgeback, Labrador Retriever, and I don't know what that last one is, a retriever? Redbone. This isn't going to deal with admixture. This isn't going to deal with the complicated situations because it was trained on pure situations, right? If we reduce the features into phenomic measures, we might be able to deal with admixture in much the same way that genomics has. I think that'd be a real advantage over just throwing everything into a black box AI. I think Aaron's found that it can focus on the wrong things, or maybe it was Nick, image background, right? If you check the attention maps, sometimes it'll be the edge of the plot rather than the thing you're actually trying to focus on. But I do think this RAG, I'm getting pretty excited about RAG. I think that's gonna be maybe a little bit of a changer from general AI. So I'm sure I didn't remember to thank everybody. Acknowledgements, thanks to everybody. Really appreciate your time and you coming here. I look forward to some tough questions and not slowing you down from going to Chimmy's.
Questions?
Great talk, thanks, Seth. You know how we usually say that we know a lot about the light side of the moon, but not much about the dark side. So in potatoes, there is a lot that happens at night. And so I was wondering about your thoughts, thinking about the future, possibly, flying drones at night and maybe even some type of spectrophotometric measures that capture other things, and if that could add quite a bit about the, improving the use of phenomics for predicting traits. For example, like in the case of potatoes, there's a lot that happens at night, and when the temperatures are lower, so the photoassimilates like sucrose would move to the tubers. We also have even the dark side, like under the ground, so it goes like way beyond. Just your thoughts about the future.
Yeah, no, I think that's a fantastic question. I would love to get some phenomic data at night. Unfortunately, we are blocked by policy, right? Flying drones at night is not something the FAA, I think there's a special exemption you can get. We're also challenged by physics because everything we do relies on passive sensing, so it's using sunlight, right? You would need some type of active sensing to get night capture. By the way, that's the reason the VIs actually work so much better than the raw bands, is because VIs adjust for things like cloud cover and the fact that sun changes intensity. In terms of underground, if you came to Lee Hickey's talk at Plant Breeding Circle, so Lee has a paper out, I think it's not been published yet, but it's online in bioRxiv, where they were actually able to predict underground traits from above ground drone measures. So, I mean, I think the sky's the limit on that, but I don't know how to get away around the FAA or physics.
Great talk. Thank you, Dr. Murray. You mentioned about collecting high-dimensional phenomic data, thinking more on the genomic side and creating population structures so we can get the most out of mapping these traits back and hopefully identifying meaningful markers to make predictions with. What kind of populations are you thinking would be appropriate to get the most out of these high-dimensional data sets, like magic populations, biparental reals? What are you thinking?
Yeah, so from what I've seen, not just in my group's work and colleagues' work, but published, the thing that's really surprising about these phenomic measures is that they aren't specific to the population they were developed in. Unlike a genomic prediction, if you develop it in one real population, probably won't work in another one. With the phenomic measures, they seem to work a lot better across stuff and environments that they were not developed on, okay? Not perfectly, but they work a lot better. So I think the most relevant population is the population you care about most. I wouldn't create a special population. But the first thing you said that I don't want anybody to be confused about is population structure is a very bad thing, because that's where a lot of your variation gets soaked into when you try to do any type of analysis. Admixed populations where you're breaking down population structure is much, much preferable. But if you're working in a potato breeding program and you've got 3 classes of potato, admixing those probably doesn't make any sense to create some type of prediction. You're probably better off creating a prediction for each of the 3 classes. So, but you don't want to throw as much diversity as you can into a GWAS population, for instance, because population structure will take everything you care about and, you know, basically make it non-useful for finding anything of interest. Did that answer your question? Okay.
Great talk. I want to preface this by saying I do believe in phenomic selection. I mean, I think it has a lot of value, but what I worry about is comparisons that are being made with genomic selection that don't make practical sense to me. And I've seen these.
Is this the Runcie?
No, I mean, well, I mean, part of the Runcie argument, but I've written a number of, I'd say, skeptical reviews for a number of people. And the argument is this. I mean, if I compare a phenomic secondary trait measured on a grain, right, like the NIRS example, I mean, to me, that's a apples to apples comparison. I can do a DNA extraction on a kernel, make a genomic prediction. I can run it through a NIRS machine. I can make a phenomic prediction. Comparing those 2 types of predictions for yield, that makes sense. But if I plant those seeds in the field and I measure the reaction norm to that environment with a drone, and then I say, oh, well, look how much better my phenomic prediction is than the genomic prediction, we're not comparing the same things here.
That's fair.
Is that a fair?
Yeah,
okay.
I think that's fair. I think that's why I tried to preface it with, you know, genomic selection is just so much further ahead, right, in terms of where it is. So the question anybody asks when a new technology comes out is, how does it compare to the old technology, right? So we need to be able to compare it to something, right? Again, in most breeding programs, I think, if I was doing genomic selection, your parent selection is different. If you're in Corteva and you have a seed chipper and you can, you know, not plant the ones that don't have favorable profiles, that's another huge rate of gain. But the way that a lot of people apply genomic selection is they plant it all out in the field and they genotype it while it's growing in the field anyway, right? I mean, that's the way most public breeders I know, I think, are doing a bulk of their stuff. And in that case, I think it is sort of an apples-to-apples comparison, right? You're still making a decision after you've planted the seeds. You're still going to be harvesting you're still going to be harvesting for yield or whatever.
Yeah, but I mean, in that case, you're actually measuring the yield. If you planted it, I mean, the way these phenomic secondary traits, for them to be a substitute for yield, the implication is that you would plant additional material that you would collect a secondary trait on, and then you wouldn't harvest yield. Because if you're harvesting yield, you don't need to make the prediction. You just measure the trait, right?
No, I think that's a big game changer I mean, if I think about how expensive and dangerous it is to drag a combine across the state, and I could have somebody out there flying a drone over it or could get satellite data in the future, right? That would be a game changer.
I think that should be quantified, because I wonder exactly what is the economics of planting the yield trial and then not harvesting it. You know, the money that you save from not harvesting it, but you also spend some money to plant it and spray it and whatever else,
Right.
which could have been used to maybe harvest more. So I think there's some interesting economics that hasn't been done yet.
I do want to step back, though, because you said secondary trait. So, I mean, this is not, in my mind, indirect selection of a secondary trait, right? What we're really trying to do, if we're getting enough phenomic information, or I don't remember who said it, but it might have been you. You said you'd like to go back and look at flowering. We'd have the phenomic data to do that, right? If you're not collecting drone data on it, you can't do that. So there's going to be a lot of other ancillary benefits that we have not conceived yet. But for now, I think the genomic to phenomic prediction comparison is probably the closest comparison to actually get breeders going, oh, there's something useful here, right? But the way back machine approach of being able to look back 10 years ago and say, oh, we couldn't measure, you know, this disease. Now we have a model that can measure this disease. Let's go back and look at our material from 10 years ago and see if we threw away something that was really disease resistant, right? You can't necessarily do that with an approach that doesn't capture the phenomics when the disease isn't present. So I think there's a lot of other ancillary benefits, but I fully understand Runcie's point. I think it's more semantics from my perspective, but we hear it from almost everybody, so I understand it's a common concern. I'm still a believer, though.
Okay, I think that's all the time we have for questions. Please join me in thanking Dr. Murray for his presentation.
Thank you.